All right. Good evening. My name is Hugh Dunstan, and I will be your host for our webinar tonight, which is focused on looking at interactions between histidine and histamine. Tim Roberts and I formed a company called Innovate in 2019 to bring to life 30 years of research investigating the underlying biochemistry of fatigue and recovery. Our research revealed 6 amino acids were lost faster through sweat and urine than other amino acids. And these same amino acids are also utilized by the body for numerous metabolic functions. For example, forming neurotransmitters and other key metabolites. We refer to these as high demand amino acids, or HDAA, and histidine is one of these HDAA. Replenishment of these HDAA forms the basis of 2 specialized amino acid supplements that can benefit general well-being, recovery from illness, and athletic performance. During the webinar, you can submit questions in the Q&A box, and I will do my best to present these questions after the presentation for discussion. Tonight, it's my pleasure to introduce Professor Tim Roberts, who has over 40 years of research experience in immunology and chronic infections. He's an Emeritus Professor at the University of Newcastle and a member of the Order of Australia. He is still active in mentoring students and publishing articles. The title of Tim's presentation is Histamine, Histidine, Confusion. Over to you, Tim.
Thanks very much, Hugh. It's a pleasure to be here for our Vitally webinar on the 23rd of October, 2024. The question we're looking at tonight is really how important is histidine as an amino acid, and does supplementing with histidine lead to an overproduction of histamine? In other words, is there a problem with histidine supplementation with some patients and some individuals? What I want to cover tonight is that on this screen, so we'll talk about histidine, one of the 6 high demand amino acids, and the fact that some proteins are very rich in histidine and therefore need a lot of histidine for their production. I want to talk about the fact that histidine is a precursor to the very important neurotransmitter, histamine. And we will have a look at the 23 different physiological functions that histamine has. Some of them: sleep, gut function, brain function, allergy, and arousal, for example. We will look at histamine intolerance or sensitivity to dietary histamine. I think that there's some variation as to the percentage of the population. Somewhere around 1% have a dietary intolerance of histamine, which is really an impaired ability to metabolize ingested histamine. And conclusion will be to look at the histidine supplementation studies and whether, in fact, there is any implication for our question at the top of this slide. Histidine supplementation lead to overproduction of histamine. So let's have a look at histidine. Histidine, is an essential amino acid that's required in large amounts for the formation of haemoglobin, and therefore also for the body to build and maintain functional erythrocytes. About 8 to 10% of haemoglobin is indeed histidine. And if we are eating food with histidine in it, meats will have about 2.7% histidine and plants about 2.3%. Therefore, there is, in fact, a need to get histidine in our diet, or indeed supplementation can be useful. Another area that I'll come to later on is skin natural moisturizing factor. And that is what keeps us looking young and beautiful. That is what is a problem in individuals with eczema. And basically, the base protein that is first synthesized in the basal layers of the skin is profilagrin. It has about 10% histidine amino acids in it. And so histidine is vital for production of skin moisturizing factor. We'll have a look at how that happens in a minute. Histidine is also required for the formation of the dipeptide carnosine, which is a combination of alanine and histidine. And that particular dipeptide is highly concentrated in brain and in muscle tissues. It seems to be an antioxidant, an anti-glycating agent, and a chelating agent. And indeed, the athletes are very keen on carnosine as a way of getting the muscles to work a little bit better. Also, histidine is crucial for the maintenance and the proper functioning of the immune system. Histidine supplementation can influence cytokine production, and these cytokines are the signaling proteins that mediate both the inflammation and the immune responses. Histidine can help modulate the balance of pro-inflammatory and anti-inflammatory cytokines that contribute to resolving inflammation. So histidine is vital in the immune system. And as I mentioned before, histidine is the precursor for the production of histamine. So let's have a look at histamine. Histamine is decarboxylated by histidine decarboxylase. Vitamin B6 is a cofactor in this. And once again, we see the importance of the vitamins. Pretty well every one of our metabolic pathways is dotted with cofactors, and these cofactors are largely minerals or vitamins. So here, vitamin B6, a cofactor for the removal of that carboxylic acid group, the COOH, from histidine to give histamine. So where does that production occur? It occurs inside cells, and these cells then generally store the histamine in high concentration in secretory granules. Most of that we know about is in the basophils and the mast cells. And if we look at the diagram down here, the mast cell is full of secretory granules that once the membrane of the secretory granule mixes with, merges with the plasma membrane, then degranulation occurs and the histamine is released. But histamine is also synthesized in the enterochromaffin-like cells lining the gut, in the lymph nodes and the thymus. We'll come back to those enterochromaffin-like cells shortly. It's also synthesized in the brain, particularly in the hypothalamus. And just take a look on the right-hand side of my screen, and you'll see there, in the presynaptic neuron, we have histamine produced and bundled up into vesicles. But that histamine is produced by histidine being transported into the presynaptic neuron. Once again, indicating the very great importance of the histidine amino acid in this role in generating histamine. And in neurotransmission, then, once again, the histamine granules are released and they go across the postsynaptic neuron. We're going to talk in a bit about the histidine receptors, and you'll see some histamine receptors. Of course, for histidine, it's an L-amino acid transporter, which transports the histidine in. But you'll notice on the presynaptic neuron, there are H3 histamine receptors, and down here on the postsynaptic neuron, H1 and H2 postsynaptic neurons. All of these are linked to a G protein meaning a guanine phosphate binding protein, and different functions by different receptors. Very important to remember that histamine has a huge range of functions, and these functions are mediated through at least 4 receptors, and there are many more receptors being discovered as we speak. So here they are, the G protein coupled receptors, the histamine receptors, H1, H2, H3, H4. They all bind histamine and bring about varying effects. And I'd like you to note that histamine is not only involved in allergic inflammation, but histamine is also involved in immunomodulation, modulation of the immune cell chemotaxis, the immune response, inflammation itself. Different receptors bring about different functions, regulation of gastric acid and secretion, and neurotransmission. So we have a histamine synthesized in mast cells, basophils, enterochromaffin-like cells, the neurons, the leukocytes, the platelets, the epithelial cells, chondrocytes, and also tumor cells. And I just want to go back there a little bit because it's as well to remember that histamine is a small ligand. Histamine binds outside the cell, binds to the surface of the cell, specifically to a histamine receptor. And that receptor is a protein that is actually going through the plasma membrane about 7 times normally, and the tail of that histamine receptor is interacting with a guanine nucleotide binding protein, a G protein. And so once the histamine receptor is activated by binding histamine, there's a change in configuration inside the cell, and the G protein next to it is activated, usually by GTP going to GDP, and then that further signals metabolic sequences to occur within the cell. Histamine itself has a quite short half-life. In some areas, it might appear to have a half-life of a couple of seconds. Other areas, the half-life is thought to be about 5 minutes. That is because histamine is broken down in 2 ways, depending on where it is. Extracellular histamine, that is histamine that might be in the food, and that's really of great relevance to somebody with histamine intolerance. Histamine in the food is broken down in the gut by diamine oxidase. It metabolizes extracellular histamine, and that will metabolize histamine from food, from alcohol, and from microbiome production in the gut. And it is a way of modulating that external histamine. If, of course, we are eating spoiled fish that has gone off, and the bacteria have converted the histidine in the spoiled fish into histamine, then histamine poisoning can occur. And this is a form of food poisoning. It has symptoms of upset stomach, headache, allergy, and sometimes even death. So huge doses of histamine can be very bad if they come in with the food, but generally the small amount of histamines in foods are handled by the diamino oxidase. If we're taking medication that inhibits the diamino oxidase, then there is the chance for the histamine to stay around much longer. Histamine inside cells is broken down by histamine N-methyltransferase, HNMT, and that's the second way in which histamine is metabolized. It relies on sufficient levels of folate as well, another B group vitamin, vitamin B12. In the diagram there, we can see the histamines coming in with as a byproduct of the food or the bacteria generating histamine. The diamino oxidase is breaking it down. So histamine intolerance then. Histamine intolerance thought to occur in about 1% of the population. It is a disequilibrium of accumulated histamine and the capacity for histamine degradation. So it will be provoked by histamine-rich food, provoked by histamines in alcohol, provoked by drugs that release histamine, or block diamino oxidase. To be diagnosed as having histamine intolerance, you must have a negative diagnosis of IgE-mediated food allergy, negative diagnosis of internal disorders, celiac disease, and so on. And the confirmation of histamine intolerance is if the individual has a good response to a low histamine food diet. And there we have the beautiful picture of what we all love to eat. But these foods are low in histamine, and if the symptoms resolve, and the symptoms are here on the right-hand side, anything from headache, migraine, dizziness, through to sneezing, rhinitis, through to tachycardia and collapsing, through to urticaria, bloating, flatulence, and so on. If the symptoms resolve, then that is a good indication of histamine intolerance. So let's now have a look at histamine supplementation studies and whether there is an indication of histamine intolerance symptoms. We've had a look at 10 separate studies and found that histidine supplementation is associated with several health benefits, some of which are supplementation is anti-inflammatory, supplementation is histidine acts as an antioxidant, better glucose regulation with histidine supplementation, better body weight management, and improved cognitive function, and improvement in atopic dermatitis. In all these studies, no adverse reactions to histidine supplementation were reported. And there was no evidence of histamine intolerance resulting from histidine supplementation. And when you get the slides, which will be available, you will be able to have these references to dig a little deeper. Let's have a look at this study by Fern and others in 2013, carried out in China. And these were obese women with metabolic syndrome. Metabolic syndrome is a cluster of conditions that give an increased risk of heart disease, stroke, and type 2 diabetes. The cluster of conditions that occur together are increased blood pressure, high blood sugar, excess body fat around the waist, that is the apple shape up here, the apple shape, and abnormal cholesterol or triglyceride levels. Apparently, and I wasn't able to follow this up too much, but this pear shape is then not linked to increased risk of heart disease, stroke, and type 2 diabetes. However, be that as it may, this study took 100 obese women aged 33 to 51 years who were all diagnosed with syndrome X or with metabolic syndrome. They were given 4 grams per day of histidine or an identical placebo for 12 weeks. And the results were startling. There was an improvement in insulin resistance, a reduced body mass index, a reduced fat mass and free fatty acids. There was a suppression of inflammatory markers and a suppression of oxidative stress. And in this study, no side effects were observed during the intervention. Adults with atopic dermatitis, adults with eczema. And here the story of profiligrant comes in. So in the stratum granulosum, we have profiligrant, a protein with very high histidine content. That protein is broken down into filigran. So filigran is a protein within profiligrant. Filigran is very rich in histidine, and filigran, as it migrates up, you'll all know that the skin is continually sloughed off. The skin skin cells at the top eventually die and are sloughed off. But what happens with profiligrant is that it's proteolyzed, it's dephosphorylated, then the filigran monomers bind to the keratin filaments to flatten the upper layer skin cells, and eventually proteases in this area break down the filigran into free-form amino acids. And these free-form amino acids on the skin are the natural moisturizing factor. The natural moisturizing factor maintains plasticity of the skin, protecting it from damage. It allows hydrolytic enzymes to function in the process of desquamation, and it contributes to optimum stratum corneum barrier function. So if all things are working nicely, then the natural moisturizing factor is formed and the skin is looking very healthy. Different situation in adults with eczema, atopic dermatitis. And in this case, 24 adults with eczema received 4 grams of histidine daily for 8 weeks. Eczema reduced by 34% over 4 weeks, and by 8 weeks by 32%. No adverse events causally related to the ingestion of 4 grams daily of histidine. And that particular study then prompted a more routine use of histidine for eczema. And a further study that I haven't gone into here of 800 people followed over 2 years, showed no adverse reactions to the histidine supplementation. Second study with eczema. This was young children who were aged 1 to 7 years with mild to moderate eczema. And this was a randomized placebo-controlled double-blind clinical pilot study. And there was a reduction in eczema of 49% at 12 weeks in those who were taking histidine. Daily dose of 0.8 grams of histidine. There was no change observed in the placebo group. But more importantly, I think, answering to the sense of this particular seminar, there were no adverse effects related to L-histidine consumption. Now, a study, again, this one from Japan, effects of histidine on mental fatigue and cognitive performance in subjects with high fatigue and sleep disruption scores. So these are individuals, high fatigue, sleep disruption scores, daily histidine intake of just over 1.5 grams showed, I haven't put in how long it went for. I apologize for that. I believe it was over 3 months, but I'd have to check on that. Results: decreased feelings of fatigue, increased efficiency while performing memory tasks, and clearer thinking and concentration. Remember, of course, that histidine has to cross the blood-brain barrier because histamine itself cannot cross the blood-brain barrier. Histamine itself, though, once it's in the brain, its function is to improve resistance to stress and anxiety. Its function is arousal and wakefulness. Its function is in appetite, drinking, and eating behaviour. We know it modulates other neurotransmitters, and it promotes brain alpha wave activity. This particular study showed a positive effect in these patients by daily supplementation with histidine. No adverse effects were reported in the study. So as we are moving along then, I think I've given you just a brief dose of the research papers that are out there with showing supplementation with histidine is very useful. Let's look at that in the broad sense. To summarize that, we know the daily requirement for histidine for a normal adult is 8 to 12 milligrams, and we need that histidine, of course, for our daily production of red blood cells, profilagrin, and so on, all of the, and of histamine. In the histidine supplementation experiments that we reviewed, doses of around 2 grams or daily of histidine improve inflammation in overweight and obese people, decrease feelings of fatigue, increase concentration and efficiency at work. Intakes of 4 grams decrease severity in atopic dermatitis disease. And there were studies done as to the toxicity of histidine, and it was shown that once you give an individual doses of 8 to 16 grams of histidine daily, then there can be some adverse effects of the histidine. But generally, the conclusion, supplementation with up to 4 grams of histidine per day provides beneficial outcomes with no adverse impacts, particularly no adverse impacts that you could link to histamine intolerance. Very, very interesting and useful to do such a review of the literature, because histidine itself, we know, is one of the 6 high-demand amino acids, and there they are: histidine, lysine, glycine, serine, aspartic acid, and ornithine. And all of that information comes from the pioneering work that Hugh Dunstan and Margaret MacDonald did, and Tim Roberts had a minimal contribution, but I'm very proud to be associated with the work Hugh has done. So it's these high-demand amino acids have been critically examined and have been peer-reviewed. The results have been peer-reviewed. So there are 6 high-demand amino acids. Why do we say that? Well, these 6 amino acids are functional amino acids, are utilized in multiple metabolic processes that I'll show you in a minute. But our research showed that they're excreted at very high rates in sweat and urine. Everybody knows about branched-chain amino acids. Well, 5 times more histidine is lost per hour in the sweat than branched-chain amino acids. And when you are losing a lot of these histidine and the other 5 high-demand amino acids in sweat, you are putting yourself in a deficit. How does that work? Well, we all know that you can sweat up to 2 litres an hour if you're exercising. In a moderate climate, up to 4 litres a day. In a hot climate, up to 14 litres a day is sweat. And you sweat, of course, to cool the body, to maintain the body temperature. We must maintain our homeostasis. But also, when one sweats, one loses sodium. And sodium is a critical element. Sodium is one of the salt was like gold in past days in Europe, because salt is so important for life. So what happens in the sweat gland? In the sweat gland, we've got a sodium concentration the same as in plasma. Histidine concentration 54 microliters. As the fluid goes up the sweat duct, 70 to 90% of sodium and chloride ions are resorbed. Why? Because in an evolutionary sense, sodium is so valuable, it has to be retained as much as possible. And what happens is there's an exchange of high amino acids and potassium, and these are pumped into the sweat while the sodium is pumped out of the sweat and retained in the body, so that final sweat concentration, 30 milliequivalents per litre, compared to 142 milliequivalents per litre in the plasma or in the sweat gland itself. And look at histidine. Histidine, very important. We need it. It's vital for production of haemoglobin, vital for production of all sorts of proteins. And two I've mentioned. Here in the sweat, we've got 1,400 micromoles per litre compared to 54. That's 20 times the amount of histidine here is lost. And I've just done this for histidine, but all of these 6 amino acids are lost in the sweat. So there's a need to supplement them. And that is why we decided to produce a product, Optimate and Electrate, as a way of supplementing these high-demand amino acids. There they are: histidine, lysine, glycine, serine, aspartic acid, and ornithine. And these high-demand amino acids are structural proteins, are metabolic proteins. They are used to build energy proteins and immune proteins. They're involved in methylation with folate, nitric oxide, and other energy systems. They're even involved in the generation of the DNA and RNA, the porphyrin rings, as I said, of haemoglobin, involved in carnosine, and they are themselves, some of them, neurotransmitters. They're involved in detoxification. And as I showed you in that last slide, they're involved in electrolyte balance, sodium resorption, saving that vital sodium ion, conserving that vital sodium ion. So in summary, histidine is one of the 6 high-demand amino acids needed daily in high amounts to maintain our normal homeostasis. It's lost at much faster rates in sweats and urine than other amino acids. It's needed for the body to produce histamine. And histamine, as I mentioned, has at least 23 different physiological functions that histamine's involved in. So the precursor of histamine is histidine, just by decarboxylation. Histidine is very, very important. Supplementation up to 4 grams per day does not result in any reported adverse responses. And our review found no reports of histidine supplementation resulting in histamine intolerance. Currently, supplemental histidine is used in a variety of conditions that I've mentioned. It's used in neurological disorders, atopic dermatitis, metabolic syndrome, diabetes, and so on. And it's also used in muscle performance during strenuous exercise. Here are our 2 products. The high-demand amino acids for building collagen and vital energy systems, the high-demand amino acids plus electrolytes for the best hydration and muscle recovery. We suggest these supplements are important because whilst amino acids are generally available from foods, highly active humans or those with chronic health issues may not receive appropriate proportions of the high-demand amino acids to meet daily requirements. The strategy is to replenish the HDAAs, including histidine, at quantities that aim to replenish what is lost during the day. Typical requirements around 2 grams a day, but that, of course, depends on the individual, the intensity of exercise, the nature of illness, etc. And with that, I will say thank you very much for listening, and over to Hugh.
Well, thank you very much, Tim. That's been really great, and I hope everybody has enjoyed that performance. I've got some... Sorry?
performance.
It was a great production. You've done an amazing job in that reviewing entity. And I must admit, before you started delving into this, we'd had a lot of people asking us about this question. The problem, can histidine supplementation lead to issues with histamine intolerance? And I think what you've done very nicely is to show that the formation of histamine is very much tightly controlled, regulated. The histamine formed is in those small vesicles, and they're released when they need to be. But what amazed me that you've uncovered, and you might like to say a few more comments about this, is just how extensive that histamine communication system is. Even with the intolerance that some people have, their whole body function relies on this histamine system, doesn't it?
Absolutely. I also was blown away that the hard part is to know just how many more histamine receptors we are going to find if we I thought I had a beautiful slide there. I don't. Why don't I? But for instance, just in terms of the gut, histamine's involved in causing the parietal cells to release their hydrochloric acid, but it's also signaling through to the vagus nerve. It's involved in 3 or 4 different ways just in the gut. And then elsewhere, it's involved in regulation of hemopoiesis. It's involved in neurotransmitter release. Histamine is just an incredible neurohormone, and I think that the first thing is histamine won't be produced unless there is histidine. Histidine's the amino acid that we need to get in our food, and so to avoid conditions of histamine deficiency, then we need the histidine. I've got a little table here of common presentations of deficiency of histamine, and it's overactive mind, agitation, confusion or memory loss, low libido, poor hearing. Oh, that must be what's wrong with me, poor hearing. Must be lack of histamine. No, I don't think so. I think it's old age. Nausea, suppressed appetite, poor healing, low stomach acid, and so on. Yeah. So just a plethora of cross reactions in the body. And that's the thing with the body, that there are so many backup systems on every aspect of the body. It's a bit like we learned when they sent up the spaceships, that they needed to have different ways of producing oxygen in case the first one failed. Well, the body is just unbelievably backed up with backup systems.
And the thing that you showed with those studies, you've got these studies that explored the use of histidine on its own for treatment of a number of conditions, and there are a number of benefits, sort of both neurological and energy driven and so forth. And I think this is really important as a central theme, that with a few essential components, the body can actually harness these to make what it needs to make when it needs to. And if these specific components are in short supply because they've been lost at excessively faster rates than the other components, then it's hard to, for example, form haemoglobin on demand if your histidine deficient. And in fact, there are a couple of studies out there that actually showed that when they deprived individuals of histidine in their diet, they became anemic. And then when they restored histidine, their red blood cell levels and haemoglobin levels came right back up to normal. So these are really important metabolites that we're talking about, aren't they?
I agree totally. I just found this slide and I thought I'd pop it up. I hope you can see it. But if we're looking at histidine, we're getting dietary histidine from proteins we eat, and we're also getting it from the dipeptide carnosine, which is histidine. But if we're not in a situation where we're not eating, but the body's still demanding histidine, then the body's going to go to the reserve bank of amino acids, which is the proteins in the muscles. The muscles are the reserve bank of amino acids. It's going to break down muscle protein to give these high demand amino acids when they're needed. And so that process is something that exactly what you were referring to. Supplementation with histidine at that time in a drink, when you're in the middle of a traumatic experience, such as recovering from burns, or you're in the middle of the fourth quarter of a game of football, then the amino acids in the free form of the high demand amino acids are going to be very, very useful.
Just while you're on that slide, Tim, it's interesting you had the source of histidine.
Oh, I've broken it now, yeah. I've broken it.
That's right. Just remember the carnosine was a source of histidine. And this is a point worth remembering because we've had quite a few people contact us who are in sports and suffering from problems with, well, trying to maintain their red blood cell levels and the hemoglobin levels, and they're vegetarian. So one of the things about eating meat is that the meat comes encased in carnosine, because that's the natural buffer, if you like, that is a fluid that actually makes the muscles work. And this isn't normally assessed when you're talking about appraising the muscle protein, but when you actually eat meat, you're gaining access to that carnosine, and that is the extra bit of carnosine that you can get if you are eating meat. And I think that's an important factor to consider, because that skewed requirement for extra levels of the histidine in the diet can come when you eat certain products, and that's just an example with the meat product.
And I feel very proud of myself to have been able to recover my presentation.
You've done well.
This program isn't terribly easy to handle. Yeah.
So
have you finished with that slide now, Hugh?
Thank you, Tim. Well done.
We'll go back to here.
I'll just see if there's some questions coming through. Is histidine something you would consider for someone with thalassemia, or someone that has shown abnormal numbers of MCV or MCH?
I think with the thalassemia should be okay. We'd have to take that one on notice, I think.
We've got some evidence when we're doing some research on horses as our animal model that there was a good response in horses that were low in hemoglobin and also low in red cell counts. After 4 to 6 weeks, they were able to restore their levels of haemoglobin and red cell counts back to a much more meaningful level. There's another question here. Tim, I know you went over some benefits of histidine supplementation and what conditions it is indicated for, but would like to know if there are anything you would consider, such as biomarkers, specific presentation of the patient that would prompt you to supplement with histidine.
I would think that it would be measurement of amino acids in the urine. Hugh, perhaps if you got some thoughts on that one? What biomarkers?
It's really hard. It leads us to a topic of possibly a future webinar, because what happens in the circulation is that the body goes to all lengths to actually maintain constant levels in the circulatory plasma. So the levels very rarely drop even after a couple of hours of intense exercise. And we've actually got a paper coming out very shortly which addresses this very issue. So it's hard to do it from a blood test. It's also pretty hard to make an interpretation from a urine test. I think if you've got evidence of low hemoglobin levels, then I would certainly give this a try. If you've got low red cell counts, certainly worth a try. You know, it's a natural product approach to actually, you know, achieving some beneficial results. People with fatigue, poor sleep, all the things that Tim's gone through in those slides, any of those symptoms, it's basically worth trying. It's difficult too, because it crosses so many areas, doesn't it, Tim? You know, all those things that you've given evidence for improvements, it makes it sound like a snake oil wonder drug, but actually these things are central to the body being able to operate properly and perform its functions.
Yeah, I'd agree with you there. And what we'll do is put something up on our blog site afterwards in relation to these questions. I think, Hugh, would be better.
Yeah. Yes, I think let's do that. So one question which we might just address, which is interesting: will histidine from dietary intake ever be adequate for therapeutic effects discussed? One of the outcomes from that is the skewed loss of these high demand amino acids. We're losing amount of sweat because they're involved in sodium resorption, as Tim has shown you, and he's also taken you through the fact that you know, we need to take in skewed amounts of some of these things because we cannot keep up with demand always. And there's some very good reasons for that. And again, we've got some publications that address this. If you are interested, just write to us and let us know. But that issue I was talking about with eating meat, where you're getting an extra load of histidine, is an important factor. And where you can't get that, then it's definitely worthwhile considering supplementation. But Tim, have you got any other just final comments you'd like to
No,
make?
I think that there are a whole lot of very healthy people who are getting enough histidine from their diet, but there are also indications that in certain situations, histidine supplementation can be very, very important. I know I'm a scientist, but equally I know that my family and Hugh's family believe very much in daily supplementation with the high demand amino acids. That's our product, but also the microminerals and the micronutrients and the omega-3 fatty acids. I think it's worthwhile reading the work of Bruce Ames, who has a triage hypothesis for amino acids, microminerals, micronutrients, and that is that the enzymes and the metabolic pathways needed to keep you alive today will be the ones that go first to the trough for these products. The other metabolic pathways will wait their turn. What does that mean? What it means is, let's take vitamin K. There are 16 vitamin K dependent enzymes, and I think 9 of them are needed for day-to-day living to keep from bleeding to death and so on. So 9 of them are very, very important. If your individual, your patient has reduced level of vitamin K, not enough to be vitamin K deficient, but reduced level of vitamin K, then those other 7 enzyme pathways that are needed to live a healthy and long life won't be getting the vitamin K. And that won't be happening because the vitamin K in an undernourished amount will only go to the enzyme pathways that are needed to keep you alive today. So that's Bruce Ames. It's called the triage hypothesis. And it is, to me, the reason why we need to supplement.
All right, well, we've run out of time. So thank you very much again, Tim. That was a great presentation. We will email copies of the presentation slides to everyone who has registered. And we'll also send links to our resource webpage, which has even more links to references, podcasts, and video interviews that may be of interest. And this webinar has been recorded, we hope, and you can visit view it again on Crowdcast or our YouTube channel, which is at HDAA Nutrients. We'll put all this information in the email. Thanks for attending tonight's webinar, and we hope it has proved a useful insight for you. So good night for now.
Thanks very much for the comments in the chat.
Yeah. All right. Good night for now. Bye.
Bye-bye.